After studying this module, you shall be able to understand what a zinc finger protein is. Analyze why nature has chosen zinc 2 as an integral component of zinc fingers. Identify by looking at the zinc site in the zinc finger the class to which it belongs. Learn about the biological functions and applications of zinc finger proteins. Zinc finger proteins are among the most abundant proteins in eukaryotic genomes. They have variable secondary structures and enormous functional diversity. All of them have at least one common motif, the zinc finger, which was first identified in Xenopus transcription factor 3A, more commonly known as TF3A. Zinc finger protein was first recognized 30 years ago in Xenopus transcription factor 3A, which activates the transcription of the 5S RNA gene in the African clawed toad Xenopus. The primary structure of TF3A contains 9 repeat sequences of about 30 amino acids, and each repeat contains 2 cysteine residues, 2 histidine residues and 3 hydrophobic residues in conserved positions. The 2 cysteine and 2 histidine residues per 30 amino acid unit form a tetrahedral coordination complex with zinc atom, generating peptide domains. These peptide domains have a typical compact folding. The DNA binding loop formed by coordination of these ligands by zinc has a protruding finger-like shape, thus the name zinc finger. TF3A has 9 zinc fingers. The characteristic sequence for each domain is tyrosine phenylalanine, X, cysteine, X, 2 to 4, cysteine, X3, phenylalanine, X5, leucine, X2, histidine, X3 to 5, histidine, where X is a variable amino acid. The domains have a characteristic beta sheet structure on the cysteine side and an alpha helical arrangement on the histidine side. The spacing of 3 to 4 amino acids between the two coordinating histidine residues is important because it places these amino acids on the same face of the helix in an ideal position for coordination to zinc. The coordination of 2 cysteine residues, which are 2 to 4 amino acids apart, with zinc induces the formation of N-terminal antiparallel beta-beta loop structure in the TF3A as shown in the figure. This proposal of structure of peptide domain in TF3A was supported by experimental data. The cysteine-histidine coordination sphere for zinc in TF3A was confirmed by EXAFS spectroscopy. Single zinc finger peptides have been prepared and purified. Such units have been found to bind one equivalent of zinc 2 or cobalt 2. Iron 2 and nickel 2 also bind to the peptide. Metal ion titration experiments have shown that zinc 2 binds 10 to the power 3 to 10 to the power 5 times more strongly than the other ions. Since zinc is spectroscopically inactive, UV visible spectroscopic studies on the cobalt complex have been done and have showed tetrahedral coordination for cobalt. CD spectra of the peptide without zinc or cobalt showed the absence of a well-defined structure. Addition of either one of the metals leads to a change in the CD spectra. Zinc is crucial for the functioning of these transcription factors. It was observed that loss of zinc in TF3A leads to a significant change in the protein conformation, as well as loss of specific DNA binding ability. This led to the conclusion that the role of zinc in TF3A was to hold the protein in the proper conformation, so that specific amino acid side chains make specific contacts on the DNA binding site. Zinc brings the alpha helix and the beta sheet closer together, forming a hydrophobic core which is important for finger stability. The stability of the beta-beta alpha domain is because of the intra-strand cross-linking provided by coordination with zinc. The structures of very small protein domains are generally stabilized by the formation of disulfide bonds or by binding to metal ions, most frequently zinc. The binding of the metal increases the thermal and conformational stability of small domains but is not directly involved in their function. Disulfide bridges can be easily cleaved by reduction in the reducing atmosphere of the cell and the nucleus. Hence, nature has chosen zinc rather than the disulfide bridges for stabilization of structure of zinc fingers. Zinc is an ideal cation for structural role in proteins because of many reasons. The natural abundance of zinc is high. It is completely non-toxic in nature. Zinc too is high in the Irving-Williams series and thus forms stable complexes, especially with sulfur and nitrogen donors. The zinc too has a filled d orbital and hence does not have any redox activity associated with it that could promote DNA damage. Zinc too can adopt a tetrahedral coordination. The tetrahedral coordination for zinc in zinc finger stabilizes it, as higher coordination numbers will require more polypeptide organization, which might present an entropic barrier to the protein folding. So, what is a zinc finger? The classical cysteine 2, histidine 2 zinc finger is recognized as the first member of zinc binding motifs. Since then, numerous other zinc binding motifs have been identified and designated as zinc fingers. These have different structures as well as functions. Initially, the term zinc finger was used to define a repeated zinc binding motif with DNA binding properties in the Xenopus transcription factor 3A. It is now largely used to identify any compact domain stabilized by a zinc ion. In most cases, zinc ions are coordinated by cysteine and histidine residues, but in some cases only cysteine is coordinated to zinc. The rapid growth in the number of zinc finger proteins necessitates their classification. They can be classified in different ways. Of the DNA or RNA binding variety, three major classes of zinc finger proteins have been established to date in eukaryotes, based on their unique and highly conserved consensus amino acid sequences. Class 1 zinc finger, also called C2H2 zinc finger. This class is also called the classical zinc finger. It comprises hundreds of zinc finger proteins that are found in eukaryotes ranging from yeast to humans. It is described as cysteine, X2 to 4, cysteine, X12, histidine, X3 to 5, histidine, where X is a variable amino acid. The two cysteine residues and two histidine residues are tetrahedrally coordinated to zinc, as shown in the figure. It has 2 to 3 beta strands in its N-terminal sequence and one alpha helix in the C-terminal half of X12H. C2H2 zinc finger proteins are nearly 1% of total mammalian proteins. These proteins bind DNA and some can also bind to RNA or protein. These proteins contain 2 or more repeating zinc finger units. GAGA factor is the only protein that contains a single C2H2 finger. Xenopus TF3A, human transcription factor SP1, mouse ZIF268 are some examples of this class. Class 2 zinc finger, also known as C4 zinc finger. This class encompasses the cysteine 4 or C4 zinc fingers. The consensus amino acid sequence for this class is cysteine, X2, cysteine, XN, cysteine, X2, cysteine, XN, cysteine, X2, cysteine, XN, cysteine, X2, cysteine, where X is a variable amino acid. The glucocorticoid and estrogen receptors belong to this class. These steroid receptors have DNA binding domain which contain 70 amino acid residues with 8 conserved cysteine residues. DNA binding domain of both these receptors show 2 zinc atoms, each coordinated to 4 cysteines. These zinc binding sites fold to form a single structural domain as shown here. The DNA binding site is located between the 2 zinc atom complexes. The DNA binding recognition site derives from the alpha helix that is part of the linking peptide region between the 2 zinc sites. Only one of the zinc base units plays a significant role in DNA recognition. The other helps in the dimerization of the protein. Class 3 zinc finger proteins, also known as C6 zinc finger. Members of this class of zinc finger proteins possess the well-conserved motif cysteine X2, cysteine X6, cysteine X5 to 12, cysteine X2, cysteine X6 to 8, cysteine. The DNA binding domain has 6 cysteine residues bound to 2 zinc atoms. These proteins are also called as zinc clusters. They have three functional domains as shown in the figure. The first and best studied zinc cluster protein is Gal4. It is a transcriptional activator of genes involved in the catabolism of galactose. The yeast gene regulatory protein Gal4 has the dinuclear zinc-2 cysteine 6 cluster. Its DNA binding domain contains a conserved sequence of 6 cysteines: cysteine X2, cysteine X6, cysteine X6, cysteine X2, cysteine X6, cysteine. The 2 zinc coordinate to the 6 cysteines in a binuclear zinc thiolate cluster as shown in the figure. The interatomic zinc-zinc distance is 3.5 angstrom. Cysteine 11 and cysteine 28 are the bridging ligands. Zinc cluster proteins are also found in a variety of other fungal organisms such as Aspergillus nidulans and Candida albicans. The motif has not been observed in either prokaryotes or in higher eukaryotes. More recently, a more systematic method has been used to classify zinc finger proteins. This method classifies them into 8 fold groups, and the classification is based on the main chain conformation and secondary structure around the zinc binding site. Each fold group constitutes proteins that share common structural features and are frequently functionally related. The different fold groups are C2H2 light finger, gag knuckle, treble clef finger, zinc ribbon, zinc 2 cysteine 6 light finger, TAS2 domain like, short zinc binding loops, and metallothioneins. The most common fold groups of zinc fingers are the cysteine 2 histidine 2 like, treble clef, and the zinc ribbon. We will now look at the biological role of zinc finger proteins. Zinc fingers are found in fungi, plants and animals. The typical number of finger repeats and the length of linker between neighboring fingers varies greatly. Zinc finger proteins have diverse roles in many fundamental cellular processes such as transcription, translation, DNA replication and repair. Thus, they participate in many biological functions such as metabolism, signaling, cell proliferation and apoptosis. They function as interaction modules that are involved in binding to DNA, RNA, proteins and small molecules like oxidizing agents, reducing agents, metals and therapeutic drugs. In zinc finger proteins, the role of zinc is purely structural. The main interactions between the protein and DNA are formed by the side chains of residues from an alpha helix which generally bind to DNA at the major groove. This is clearly depicted in the figure. The figure shows a classic zinc finger PDP entry 1ZNF with 25 amino acids. Except the 2 cysteines, 2 histidines and the 2 hydrophobic amino acids phenylalanine and leucine, the other amino acids face outwards. The top portion of the figure shows the above mentioned classic zinc finger bound to DNA with 3 such fingers, and the lower portion the protein without the attached DNA. The interactions of zinc finger proteins with DNA can be understood using the C2H2 zinc fingers. C2H2 fingers bind DNA segments with a specific affinity because of the 4 amino acid residues present in the alpha helix of the finger, in the N-terminal part of the alpha helix. These residues participate in DNA recognition by interacting with hydrogen donors and acceptors exposed in the major groove. The mode of binding of C2H2 finger to DNA was first determined by studying the protein ZIF268. This protein contains 3 zinc fingers. The alpha helical portion of each finger fits in the major groove of DNA. The binding of successive fingers causes the protein to wrap around the DNA. Crystallographic analysis of zinc finger ZIF268 DNA complex formed by the interaction of 3 zinc fingers with DNA revealed that the 3 zinc fingers bind in an equivalent manner to a segment DNA 9 base pairs long in the major groove, so that each zinc finger contacts 3 base pairs with no gaps between the 3 sites. The linkers between the adjacent fingers play little or no role in DNA recognition. Let us now briefly look at zinc finger engineering. In recent years, considerable effort has been done in the field of designer zinc fingers. Designer zinc fingers are artificially zinc fingers, usually C2H2 zinc fingers, with different specificities. They can specifically target unique binding sites within the human genome and thus have important applications as probes and in human gene therapy. Efforts to increase the zinc finger DNA binding affinity can be done in 2 ways. One is to increase the number of zinc fingers. The second way is to change the linkers for connecting 3 finger modules. However, there is not much improvement in affinity when more than 3 fingers are connected by TGEKP linkers. So, more focus is on the latter approach. The summary of what we have learned in this module is: zinc finger is any compact domain stabilized by a zinc ion. Zinc has a structural role in zinc finger. Zinc holds the protein in the proper conformation. The 3 major classes of zinc finger proteins based on the unique and highly conserved consensus amino acid sequences are class 1 zinc finger C2H2, class 2 zinc finger C4 and class 3 zinc finger C6. Zinc finger proteins have also been classified into 8 fold groups, and the classification is based on the main chain conformation and secondary structure around the zinc binding site. Zinc finger proteins have diverse roles in many fundamental cellular processes such as transcription, translation, DNA repair and replication. They bind to DNA, RNA, proteins and small molecules. Designer zinc fingers are artificial zinc fingers, usually C2H2 zinc fingers, which target specific DNA sequences.